Comprehensive overview of DNA. DNA, deoxyribonucleic acid, is the molecule that stores and transmits genetic information in all known living organisms and many viruses. It plays a crucial role in heredity, development, and biological function. Below is a detailed breakdown of DNA's properties, functions, modifications, interactions, evolution, and technological applications. 1. Properties of DNA. DNA is a double-stranded helix composed of nucleotides. Chemical composition, nucleotides. The building blocks of DNA, each consisting of a phosphate group, a deoxyribose sugar, a nitrogenous base. Adenine, A, thymine, T, cytosine, C, guanine, G. Base pairing, A binds with T and C binds with G via hydrogen bonds. Double helix DNA strands run antiparallel and are held together by complementary base pairing. Physical properties: stable under normal conditions but can denature separate under high heat or acidic conditions. Can replicate itself through semi-conservative replication where each new DNA molecule retains one original strand. 2. Chemical modifications and altered DNA packaging. DNA can be chemically modified and packaged in different ways affecting gene expression and function. Chemical modifications: DNA methylation, addition of methyl groups to cytosine bases, often silencing genes. Histone modification, DNA wraps around histones, proteins, and chemical changes to histones, acetylation, methylation, affect gene expression. DNA damage and repair, UV light, radiation, and chemicals can cause mutations. For example, thymine dimers, which are repaired by DNA repair enzymes. DNA packaging and chromatin. Euchromatin, loosely packed transcriptionally active DNA. Heterochromatin, densely packed transcriptionally inactive DNA. Chromosomes, highly condensed DNA structures during cell division. 3. Biological functions of DNA. 1. Genetic information storage. DNA encodes the instructions for building proteins and maintaining cellular function. 2. DNA replication. Cells duplicate DNA before division to ensure genetic continuity. Enzymes involved: DNA helicase unwinds the double helix, DNA polymerase synthesizes new DNA strands, ligase seals gaps between newly synthesized DNA fragments. 3. Gene expression, transcription, and translation. Transcription: DNA is copied into messenger RNA, mRNA. Translation: mRNA is read by ribosomes to assemble proteins. 4. Mutation and genetic variation. Mutations in DNA sequences drive evolution and can cause genetic disorders. 4. Interactions with proteins. 1. Structural proteins. Histones package DNA into chromatin. Topoisomerases prevent DNA from overwinding during replication. 2. Enzymes and regulatory proteins. DNA polymerase synthesizes new DNA strands. RNA polymerase reads DNA to make RNA during transcription. Transcription factors bind to DNA to regulate gene expression. 15. Genetic recombination. 5. Genetic recombination. Genetic recombination is the exchange of genetic material between DNA molecules, increasing genetic diversity. Homologous recombination, exchange between similar DNA sequences, for example during meiosis. Non-homologous recombination, DNA segments are inserted or rearranged without similarity. Horizontal gene transfer, bacteria can exchange DNA through conjugation, transformation, or transduction. 6. Evolution of DNA. 1. RNA world hypothesis. DNA likely evolved from RNA, which was originally the genetic material of early life. RNA is still central to many biological processes, for example, ribosomal RNA. 2. Early evolution of DNA. DNA replaced RNA as genetic material because it is more stable and allows for later genomes. Over time, DNA evolved repair mechanisms to fix mutations, increasing genetic fidelity. 3. Evolutionary changes in DNA, gene duplication, creates new genetic material for evolution. Pseudogenes, non-functional remnants of genes. Mobile genetic elements, transposons, jumping genes, move within genomes. 7. DNA uses in technology, genetic engineering and biotechnology. CRISPR-Cas9 used for gene editing. Recombinant DNA used to produce insulin, vaccines, and GMOs. 2. DNA sequencing used to read genetic information for medicine, ancestry, and research. 3. DNA forensics. DNA fingerprinting helps identify individuals in crime investigations. 4. Synthetic biology. Scientists are designing synthetic DNA to create artificial life forms. 5. DNA storage. DNA can be used to store digital information, offering high-capacity data storage. 8. History of DNA. 1. Early discoverers. 1869, Friedrich Miescher discovers DNA, initially called nuclein. 1928, Frederick Griffith shows that genetic material can be transferred, bacterial transformation. 2. Identification of DNA as genetic material. 1944, Avery, MacLeod, and McCarty prove that DNA carries genetic information. 1952, Hershey and Chase confirm DNA, not protein, is the genetic material. 3. Discovery of DNA structure. 1953, James Watson and Francis Crick, using Rosalind Franklin's X-ray images, proposed that double helix model of deoxyribonucleic acid. 4. Advances in molecular biology. 1977, DNA sequencing developed by Frederick Sanger. 2003, completion of the Human Genome Project, sequencing all human DNA. Conclusion: DNA is central to life, governing genetic inheritance, evolution, and cellular function. It has revolutionized science, medicine, and technology, and ongoing research continues to unlock its potential